Morning everyone, myself Sheikh Ismail Ali Basha, Department of Botany, the 12th semester Unit 2, Embryology of Angiosperms. The structure of anther, the male reproductive part of the flowering plant, has stamens. These stamens are filamentous, the structures that support the anther that produce the pollen grains. These anthers have two lobes and they are termed the bilobed anthers. The transverse section of the anther helps to distinguish this bilobed structure. The two lobes of the anther are formed by the septum. Each lobe of the bilobed anther has two theca and two microsporangia. There are hundreds of microspores produced by each microsporangium. The anthers are lobed so that they can accommodate more spores. The filamentous stamens support the knob-like anther on its top. A typical anther is always a bilobed and a dithecous structure. Theca is a chamber on the anther lobes. Usually each lobe has two theca and is hence named as a dithecous. The transverse section of the anther shows two microsporangia in each lobe. Thus they have four microsporangia in total. The microsporangia or the pollen sac consist of the pollen grains. It is the anther structure. The structure of anther wall. It is consist of four layers. The epidermis, it is the outermost layer and is a single layer that is a thickened fibrous matter whose function is to protect the pollen sacs. Endothecium or the inner epidermis is a single layer for the radially elongated cells that develops the cellulose with the pectin and lignin fibers thickening around the sac which holds the two together and prevents it from splitting or bursting open. The middle layer is a thin-walled cell of 3 to 4 layers situated just below the endothecium nourishes the microspore for facilitating pollen production. The fourth layer that lies closest to the pollen sac is nutritive and provides necessary nutrition for all pollen divisions or pollen generations. The types of tapetum. A tapetum is the deepest layer of the anther which encompasses sporogenous tissue. The sporogenous tissue produces the microspores that supply enzymes and the nutrition for pollen development. Tapetum is a mostly multinucleate due to the mitotic cell division. Abnormal cell division can also lead to the tapetum polyploidy. Types of tapetum: secretory or glandular tapetum. The surround the anther loculi. This type of secretory tapetum is more common in angiosperms. Amoeboid or plasmodial tapetum. After the degeneration of the tapetal cell wall, a multinucleate structure along with the cell protoplast called the plasmodium tapetum is formed. The microsporogenesis. The microsporangia or sporangia that produce the microspores that create the male gametophyte when germination occurs. The microsporangium is an important part of the gametophyte, the reproductive organ of the plants where pollen grains are formed. Microsporogenesis takes place inside the flowering plants. The microsporangia, the spores are a plant's reproductive organs. Microsporogenesis is the process by which microspores are generated inside pollen sacs, also known as a microsporangia. The structure of microsporangia. The microsporangia are bilobed structure found at the end of the long filament like stamen that function as pollen sac in plant's anther. Microsporangium has a circular form with a 4-layer encircling it. When the epidermis matures, it stretches and then falls off a specific pattern. A fibrous endothecium is a type of cell with fibrous ends. The layers in the middle are the most important. The cells in the intermediate layer of the fully developed anther normally break down. A tapetum layer with the dense cytoplasm, the uninucleate, binucleate or multinucleate layers. The microsporogenesis. The microsporogenesis is the meiotic or reduction division process that produces the pollen grains inside the pollen sac of the flowering plants. In other words, the microsporogenesis happens when meiosis or reduction division occurs in the nucleus each microspore mother cell resulting in 4 haploid nuclei. Diploid sporogenesis cells transform into microsporocytes during the microsporogenesis process which divide to produce 4 haploid microspores during the meiosis. Microsporogenesis is finished when individual single-celled haploid microspores arise from each diploid meiocytes, a tetrad of a 4 haploid. This diagram. Structure of ovule. The ovule is a multinuclear oval-shaped structure which originates from the placenta and lies inside the chamber of the ovary. Each chamber may consist of one or more member of ovules. A fully mature ovule consists of the stalk or funicle on the body. One end of the funicle is attached to the placenta and the other end of the body of the ovule. The point of attachment of the nuclei with the body is called helium. The nuclei sometimes extend up to the base of the ovule and the rings thus formed is called raphe. This is the diagram. The megasporogenesis. Introduction. The process of development of megaspores from the megaspore mother cells is known as a megasporogenesis. The megaspores are developed into the female reproductive organs. Development. A layer of sporogenous tissue is present in the ovules of the female flowering plant. The megaspores mother cells are developed from the sporogenous tissue. It is large diploid cell with prominent nucleus in dense cytoplasm. The MMC undergoes meiotic nuclear divisions to produce 4 haploid megaspores. The megaspores are arranged axillary called the linear tetrad. Out of the 4, only one megaspore is functional and the rest 3 degenerate. The megaspores at the chelazal end are functional and 3 towards micropellet regenerate. The one functional megaspore divides mitotically to form the embryo sac, a process called megagametogenesis. It divides multiple times to produce 8 haploid nuclei for the embryo sac, which is called as a megagametophyte. The types of embryo sac development: monosporic, bisporic, tetrasporic. Monosporic. In the majority of the angiosperms, the embryo sac develop from the one functional megaspore while the other 3 generate. Example, Oenothera polygonum. Bisporic. In this type of 2 of the megaspores takes part in the embryo sac development. Example are Allium, Trillium, Siscula. Tetrasporic. In this type of all 4 megaspores take part in the embryo sac development. For example, Peperomia. Pollination is the act of transferring pollen grains from the male anther of a flower to female stigma. The goal of every living organisms, including plants, is to create the offspring from the next generation. One of the ways that plants can produce offspring is by making seeds. Seeds contain the genetic information to produce a new plant. Pollen-pistil interaction. All pollination do not lead to successful fertilization because for successful fertilization the pistil of a flower has to recognize the pollen of the same species. Therefore, the interaction between the pollen grains and the stigma needs to be understood properly. Once the compatible, the pollen is recognized and accepted by the pistil, events for the fertilization proceed. The pollen which is incomplete is rejected by the pistil so that process the fertilization does not proceed. Upon reaching the pistil, the pollen grains release the chemicals which are recognized by the pistil. This pollen-pistil interaction results in the acceptance of the compatible pollen. The events of pollen-pistil interaction proceed as follows. The leading of true pollen on the compatible pistil, germination of pollen and formation of pollen tube where pollen grains release their contents. The pollen tube grows through the styles of the pistil towards the ovary, the entry of male gametes into the ovule and then to synergy. Fertilization. After pollination is deposited on the stigma, it must germinate and grow through the style of the reach of the ovule. The microspores or pollen contain 2 cells, the pollen tube cell and the generative cell. The pollen tube cell grows into the pollen tube through which the generative cell travels. The germination of the pollen tube requires water, oxygen and certain chemical signals. As it travels through the styles to reach the embryo sac, the pollen tube growth is supported by the tissues of the style. During this process, if the generative cell has not already split into the 2 cells, it now divides to form 2 sperm cells. The pollen tube is guided by the chemical secreted by the synergids present in the embryo sac. It enters the ovule sac through the micropyle. Of the 2 sperm cells, one sperm fertilizes the egg cell, forming a diploid zygote. The other sperm fuses with the 2 polar nuclei, forming a triploid cell that develops into the endosperm. Endosperm is a tissue present inside the ends of the most angiosperms following double fertilization. It is rich in carbohydrates, proteins and lipids and provides the nourishment to a growing embryo. Types of endosperms: nuclear endosperm, cellular endosperm, halobial endosperms. Nuclear endosperms, primary endosperm nucleus undergoes several meiotic division without cells as well formation, thus a free nuclear condition exists in the endosperm. Example, Coccinea, Klebsiella, Arachis. Cellular endosperms, primary endosperm nucleus divided into 2 nuclei and it is immediately followed by the wall formation. Halobial endosperm, the primary endosperm nucleus moves towards the base of the embryo sac and divides 2 nuclei. Cell wall formation takes place leading to the formation of large micropylar and small chelazal chamber. The nucleus of the micropylar chamber undergoes several free nuclear divisions, whereas that of chelazal chamber may or may not be divided. Example, Hydrilla vallisneria. Endosperm biological importance. The nutrient reserve. The endosperm serves as the storage site of a nutrient like a starch, protein and oils which are vital for the germinating seedling energy needs. Embryo support. It provides the nourishment to the development embryo, supplying the essential resources required for growth and development. Germination energy. The stored reserve in the endosperm provides the energy necessary for the initial stage of germination when the embryo start to grow. Seedling growth. Endosperm nutrients are used by the germination seedling until it can establish functional leaves and roots, the photosynthesis and nutrient absorption. Early root development. The endosperm nutrients support the early development of the root system, aiding in anchoring the plant and absorbing water and minerals. Trophic function. It acts as a trophic tissue, enabling the embryo to continue development even when conditions for the photosynthesis are not yet optimal. The development of dicot. The zygote divides transversely to form a two-celled proembryo. The cell towards the micropyle is known as the basal cell and the other known as the terminal cell. The basal cell undergoes several transverse divisions for form a long suspensor. The terminal cell divides longitudinally twice for 4 cells. The 4-cell stage of terminal cell is called quadrate stage. The 4 cells of the quadrate stage now divided transversely form a octant stage of 8 cells arranged in 2 tiers of 4 cells. The lower tier gives rise to the stem, tip and cotyledons, while the upper tier is meant for the formation of hypocotyl. This is followed by periclinial division in the octant cell to give rise to the 8 outer cells and the 8 inner cells. The 8 outer cells form the dermatogen which divide anticlinally and develop into the epidermis. The inner cell form the peribellum and pleroma. The cortex develop from the peribellum and stele from the pleroma. The basal cell divides several times to form a long suspensor of 6 to 10 cells. The lowermost cell of the suspensor nearest to the developing embryo is known as a hypophysis. The hypophysis by repeated division gives rise to the root cap, epidermis and cortex of the root. The further enlargement of the hypocotyl and the cotyledon result in the curvature of the cotyledons. The embryo at the thin stage appears as a horseshoe-shaped structure. In the mature embryo, the stem tip is terminal and the 2 cotyledons occupy the lateral position. The end.